supporting such academically enriching initiative. Till now, we have attended sessions under the Faculty Development Program on Artificial Intelligence as a catalyst for excellence in pharmaceutical education, research, and innovation. During this session, we learn about the application of AI in academics, excellence, literature review, and research enhancement. We explore AI tools for creating infographics, flowcharts, graphical abstracts, and presentation, such as Napkin AI, Create AI, and Notebook AI. We are also gained insight into AI and ML for non-programmers. The session covered assessment tools like Question Bell for MCQ and quiz generation with Bloom's taxonomy, as well as Zotero for reference management. Overall, the session enhanced our understanding of integrating AI tools into pharmaceutical education, research, and innovation. Now, I would like to introduce today's esteemed resource person, Dr. Umang Shah sir. Dr. Umang Shah sir is a distinguished academician, researcher, and mentor. Dr. Shah is currently serving as an associate professor at Ramanbhai Patel College of Pharmacy at Gujarat, with over 18 years of experience spanning academics, research, and industry. His area of expertise include drug design and discovery, medicinal and pharmaceutical chemistry, and anti-cancer drug development, with significant research contributions in cell culture-based assays and anti-cancer drug screening, making him an ideal expert for today's session. Dr. Shah has an impressive and over 1,000 citations. In addition, he is a well-known academic author and speaker, having published GPAT preparation books with Pearson Education and contributed book chapters to Elsewhere and Taylor and Francis, and delivered 40 plus invited talks at national and international platforms. His contribution has been recognized with prestigious awards, including Young Researcher Award and Best Talent Award. Without a further delay, I now invite Dr. Umang Shah to kindly begin today's session on application of cell culture techniques in anti-cancer drug screening and share his valuable knowledge and practical insights with us. Over to you, sir.
Am I audible?
Yes, sir.
Yeah.
Yes, sir.
Okay. So thank you very much, madam, for nice introductions. So first of all, I would like to thanks to Rajendra Gode Institute of Pharmacy as well as Rajendra Gode College of Pharmacy, as well as convener of the FDP program, Dr. Pramod sir, Dr. Hari Gopal sir, principals and management. Also, I thank to Vrushali madam, Nikita madam, Neha madam, and Yogesh sir, coordinator of this event, and Rahul sirs and other advisory committee member for inviting me on to talk on cell culture technique on the screening of the anti-cancer drugs. Okay. So I think this is somewhat different topic than you discussed in last 2 days, but it is quite interesting. I have taken all basic concept, all basic assays. What are the requirements needed to establish cell culture laboratories? What are the equipments required? How to procure medias and everything. I have mentioned each and everything, but it is very basic presentation. I will also show the some results which is getting from cell culture techniques. So why this technique having a importance nowadays? Because when you study on animals. So to produce the cancers, particular cancers in animals, so it will take more times. It is quite complicated technique, kind of expertise is required. So compared to these animal studies, this technique is quite useful for anti-cancer screening because cell lines are available. Uh, Generally, I procure the cell lines from NCCS Pune. And if you want preliminary studies, you can easily perform the preliminary studies, and within 15 days, you will get the data. So let us start with the presentation. So what is cell cultures? Okay. So basically cell culture, which refer the removal of the cells from the animals or plants or their subsequent growth in favorable artificial environments. Okay. So how cell line is developed? So initially they have taken the cells from animals. They have also taken the cells from humans. Okay. So say for example, person is suffering from breast cancer. So they have removed the breast tissues from these cancerous cells. And from these tissue, they are performing the primary cultures and they isolate the cells and by providing an artificial environment and they are growing the cells and later on it is called as a cell lines. Okay. So primary cultures here I mentioned the stage of the culture after cells are isolated from the tissues and proliferated under appropriate conditions until they occupy the available substrate. Occupy the available substrate that is called confluent. Confluence means whenever you are growing the cultures and until and unless you are getting 90% of the cells are grown. So they are called confluent flask. Okay. So once your flask is confluent, then you need to subculture. Subculture means passaging. So whenever you are getting the cell line from NCCS, so they have mentioned PN number. So it is nothing but it is passage number. सो पसाद नंबर क्या होता है कि सबसे पहले जो टिश्यू में से सेल आइसोलेट किए उसके बाद उसका कितनी बार सब कल्चर किया है दैट इज कॉल्ड पसाद नंबर. Okay, so whenever cells are confluent, we need to subculture. So first subculture kiya, that is called Passad number 1, and then subsequently you are growing the cells in your laboratories, and meanwhile Passad numbers are increasing. So in cell line study, if passage numbers are less, that means cells are morphologically good. They are giving a better result. Okay. Then after first subculture, the primary culture that is become a cell line or we can say subclone. So this is called cell line. Now for growing of the cells, various media is required, various supplements is required, just like growing of our cells. Okay. So for artificial environment we need to create. So for that we are using various media and each cell lines having a different kinds of media. I'll show you which medias are required. And these media contain nothing but various amino acids are there, certain carbohydrates, more specifically, they are using glucose solutions. Certain vitamins are also there and minerals are also there. For growth factor, they are using fetal bovine serum. And in some cases, they are also using insulin as a hormone. And we know that cells are growing in CO2 incubators. So generally 5% CO2 at body temperature means 37-degree temperatures. We need to grow the cells. So why CO2 is required? Because it maintains the biocarbonate and carbonic acid balance. Okay. And they are not convert your media to become alkalines. Okay. So if CO2 level is not maintained, then they are directly affect the cell growth as well as the visibilities. Now, if I am talking about the types of cells, so basically there are two types of cells are there. One is anchor dependent and another is suspension culture. Okay. So anchor dependent means once you grow the cells, so with T25 flask, it is attached to the flask and further it is not moving. And with the flask surface, they are growing well. So That is called anchor dependent as well as adherent cell cultures. एंड दूसरा जो टाइप है दैट इज कॉल्ड सस्पेंशन कल्चर. तो उसमें सेल सस्पेंड रहते हैं, दे आर नॉट गोइंग टू अटैच ऑन फ्लास surface. So there are two types of cells are there. But in our laboratory, generally we are working on adherent cells because majority of the cells are adherent. Now there are basically three types of morphology we have seen in cell culture techniques in cells. So first image you can see it is a fibroblastic cells. So basically they are bipolar as well as multipolar and also having a elongated shape and grow attached to the substrate. That means they are growing as a adherent cells. Second image is epithelial cells. So they are basically polygonal in shape and regular dimensions are there and they are also considered as a adherent cells. And last cell, which is called lymphoblast like. So they are generally spherical in shape and usually they are growing in suspension. So they are called suspension culture. Now, to establish a cell culture laboratory, so certain instruments you need. So first and prime requirement is cell culture hood or हम उसको बोलते हैं biosafety cabinets. So biosafety cabinet are having a two types where laminar airflow are there and two types are there, vertical flow and horizontal flow, where airflow are verticals. So according to my experience, vertical flow laminar air biosafety cabinets are very good to avoid the contaminations. Next is incubators. So first is image of incubators. So in incubator, generally, as I mentioned, we need to maintain the CO2 level at 5% and the temperature which is required for cell growth is 37 plus or minus 0.5 degree and 95% humidity. So just last tray of the incubators. So we need to supply autoclavable water in incubators and we need to monitor the water level whenever you are using CO2 incubator. So water level is also there to provide its optimum condition. Next, we require cooling centrifuge. So why there is a requirement of cooling centrifuge? So whenever your cells are adherent cells, so by adding trypsins, so trypsin is a proteolytic enzyme. So after trypsinizations, your cells are coming into the media and whatever the morphologies are there, once you trypsinize the cell, it is converted into the round shape. And after that, you need to centrifuge your cells. And after 10 minutes of centrifugation at 200 G, G means gravitational force. So you will get the cell pellet. Okay. And then you can count the cells by Newbar chambers. How to count, I'll show you. So for that purpose, cooling centrifuge is required. Water bath is also required because whenever you are using any media, so some medias, it should be stored at freezing conditions. Other part is also there in complete media preparation. We need to use antibiotic, we need to use FBS. So these kind of chemicals, it should be stored in minus 20 degree. So whenever you need to prepare for complete media, so these all medias, it should be at room temperature before preparing complete media. So for that, water bath is required. Minus 20 and simple freeze are required, as I said. So recently we purchased cell counter. It is automated cell counter for corning. So we just keep a 10 microliter of cell suspension and 10 microliter of trypan blue dye. So basically, trypan blue dye is used to differentiate the live and dead cells. So we just mix together and on slide, we just put the slide in these equipments. So directly we are getting the number of live and dead cells. So while putting the assay, we require the live cells. Dead cell, we need to omit it. So this is a perfect instrument. You will get perfect result by using automated cell counter. Deep freezer as well as liquid nitrogen container are required to maintain the cells. Okay. So currently in our laboratory, 4 to 5 different cell lines are, we are generally in growing phase. Students are working on those cell lines. But once work is completed, then we need to store the cell lines. So how to store, how to prepare freezing media, I'll let you know in later on slides. But generally, we are storing the cell lines in -80 deep freezers. And whenever we regrow it, so we remove the vial and we can regrow it. So once you regrow the cells and once you store the cell, so it would be suggested that within 3 months you need to regrow it. Okay. Otherwise, sometime what happen, it is not growing well. Next important uh, instrument or equipment we can say, that is called autoclave. Okay. So whatever the tips you are using, whatever the append drop tube you are using, so each and everything should be autoclave before the using. In market already sterilized tips are there, but it is advisable to perform re-autoclave for those tips before using or before use in cell culture technique. And once it is autoclave, so in the tip box there are formation of small droplets. So for that we are using dry heat sterilizers, and whatever the droplets are there, within 1 to 2 hours at 60-degree temperature, the all droplets are removed. The next instrument is inverted microscope. In our lab, we have an inverted microscope with fluorescence facility. So we are also performing various fluorescence dye-based assay. So that is required because in paper they are asking for morphology changes whenever you are treated your investigated drug to various cancerous cells. So compared to normal cells or compared to control, if your drug is quite effective on cancerous cells, so the cell's morphology has been changed. I'll show you the example. Then generally plastic wares are used. So generally we are using plastic wares from Nest, Corning and Tarson's. Now procurement of cell lines. So there are various ways to procure the cell line. Among them, one is American type cell cultures. So it is largest and most renowned cell line repository in the world, and they provide most authenticated cell line for research purposes. But the costing of these 80 cc cell line is quite high. Per flask, they are charging more than 50,000 rupees. Next is European collection authenticated cell cultures, then Japanese collection of research bioresources cell bank, Korean cell bank, and in India, we are generally procuring the cell line from NCCS Pune. Before 3 to 4 years back, they are only giving one flask of the cell line at the price of 500 rupees, but nowadays they are charging 4,000 rupees plus GST. So total cost for one flask is 4,720. Okay. So for different cell lines, we need to require different medias. So according to my experience, MEM and DMEM media you can procure from high media. Cells are well growing in those kind of media. Other media like L15, RPMI, F12K, those kind of media, I suggest that you should procure from Gipco. It is a brand of Thermo Fishers. Okay. So in this slide, I have mentioned various cell lines and various species from where it is obtained, and kind of media which is used to growing the cells, and supplement they have mentioned that 10% FBS means fetal bovine serums. Okay. Sometimes they have mentioned that with FBS you need to add glutamine, you need to add other kind of chemicals. So how we can know which kind of media should be suitable for cell line and what other supplements we need to add? So once you procure the cell line from NCCS Pune, so with cell line they are giving you a one certificate, one feedback form, and one protocol. So in protocol they have mentioned that which media you have to use, what percentage of FBS you should use. So in generally, majority of the cell line, 10% FBS we are using, it is act as a growth media. Okay. So with protocol, everything is mentioned. So based on protocol, we need to use the media. Now, whatever the media we are procuring from the market, so this is not called complete media, because in media we need to add certain other supplement for the growing of the cells. Okay. So in generally, we are preparing the media for in a volume of 50 ml. So in volume of 50 ml, we need to take 50 ml sterile tubes, then FBS, that is act as a growth serum. Then antibiotic and antimycotic solution is also required. So for 50 ml, generally we are adding 500 microliter of antibiotic antimycotic solution. And this is a standardized protocol in cell culture technique. Then HAPAS buffer we need to add. So what is role of HAPAS buffer? So once you are growing the cells in incubators, and sometime it happen due to the electricity breakdown, so your incubator is going to be stopped. So in these case, if you added HAPAS buffer, so that is also activate the carbonate buffer systems, and which will help to growing of the cells, and your cells are remain intact. Okay. And whatever the media is there in protocol, you need to add it. So FBS, if we consider 10%, so for 50 ml, we need 5 ml FBS, 500 microliter antibiotic solution, 1.25 ml HAPAS solutions, and remaining media. Mark up the volume up to 50 ml, and then through syringe filter, we again need to filter it, because whatever the medias and reagents are there, they are already sterile. But in laboratory, you have open it, so there may be a chances of contamination. So for that, we need to again filter through 0.22 micron filter, and whatever the filtered media are there, that we need to use for further assay. This is freezing media preparation. So generally we are preparing 10 ml of freezing media, because for storing of one vial, 1 ml of freezing medias are required. So in freezing media, you need to take complete media which is prepared previously. Then add 2.5 ml FBS, because it is a growth factor, and your cells are completely stored at minus 80 degrees. So it should be alive. So for that, FBS level, it should be more. And DMSO and glycerol, which is act as a cryoprotectant. So we add in 0.5 ml, and then you can freeze the cells by using this freezing media. Now, what are the role of trypsin in cell cultures? So as I said, trypsin is a member of serine protease family enzymes, and which is frequently used in cell culture technique to detect the cells. Okay. So previously, before 5 to 10 years, whatever the trypsins, whatever the PBS we are using, phosphate buffer, so it is available in powder form. But nowadays, ready-made trypsin solutions are available. It is called trypsin EDTA solutions. Okay. So the role of trypsin is it is used to dissociations of the cells. Okay. So whatever the cell lines we are using, it is adherent cell lines. So to detect the cell, we need to add trypsins. So what are the concentration of trypsin? So if you grow the cell in T25 ml class, so 1 ml trypsin is required. And if you are growing the cells in T75 class, so 3 ml trypsins are required. So initially, what are the procedure for trypsinizations? So you need to remove the flask from incubators, then remove all the media from the T25 flask, wash the flask with PBS buffer twicely, remove the PBS buffer, and then add 1 ml of trypsin in the T25 flask, and keep the flask in CO2 incubator for a period of 5 to 7 minutes. Trypsinization, it is again complicated procedure. If you keep the flask in incubator with trypsin for more than 10 minutes, so your cells are automatically degrade. Okay. It is not used for the further assays. So make sure that within 5 to 7 minutes, you need to remove the flask and then after mechanically you just tap the flask. So whatever the cells are there, whatever the morphologies are there, that is totally changed. You can see here, this is MCF7 cell line, and after trypsinizations, all cells are become round shape. And you can see in the microscope, they all are floating. Okay. So once it is floating and they are in cluster form, so after minor tapping the flask, after providing the mechanical pressure to the flask, so your each and every cells are detached with each other, and you can see the each cell in round shapes. So in cell culture technique, we are using trypsin for cell detachment. Now, how to check how many live and dead cells are there? Okay, so for that we are using two techniques. One is Newbar chamber, which is generally used in HAP practicals, and another we are using automated cell counter. So in Newbar chamber, you observe this kind of 16 squares are there. Okay. So once you add the type blue dye, so for cell counting, we need to take 10 microliter of cell suspension and 10 microliter of trypan blue dye and mix together, and you just keep 10 microliter of this mixture in Newbar chamber. So after watching under microscope, you can see these kind of cells. So these kind of cells are called live cells. तो उसमें क्या होता है कि लाइव सेल्स जो होते हैं उसका जो बॉर्डर है सराउंडिंग वो स्टेन होता है, और जो डेड सेल होता है वो का सेल स्टेन हो जाता है, मतलब बीच में जो व्हाइट स्पॉट है वो नहीं दिखता. They are called dead cells. So what we need to how to count? So first in 16, तो इसको अपन बोलेंगे ये A हो गया, then B, C and D. ये चारों जो 16 स्क्वायर है उसमें से चारों में सेल काउंट करने हैं, कितने लाइव सेल है कितने डेड सेल्स. सपोज आपने काउंट किया एंड टोटल आता है 500 सेल्स. तो इसमें 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 और इसमें ABCD चारों साइड पे 500 सेल है, तो उसका आपको एवरेज निकालना है. तो डिवाइडेड बाय 4 करोगे तो वी आर गेटिंग एवरेज 125 सेल. इनटू 2 करना है क्योंकि हमने 10 माइक्रोलीटर ट्राइपन ब्लू लिया था और 10 माइक्रोलीटर सेल सस्पेंशन लिया था, और इनटू 10 डाइल्यूशन फैक्टर. तो 1 माइक्रोलीटर हमारे पास 1 ml सॉल्यूशन है, तो उसमें 1 माइक्रोलीटर में इतने सेल्स है. तो हमको क्या करना है, 2500 सेल्स है. तो फॉर ईच वेल, सपोज अपन एमटीटी ऐसे कर रहे हैं तो 96 वेल प्लेट में ईच वेल वी रिक्वायर अप्रोक्सीमेटली 10,000 सेल्स. सो 10,000 डिवाइडेड बाय ये जो सेल्स आते हैं वो करोगे तो यू विल गेट द वॉल्यूम. मतलब आपका जो 1 ml सेल सस्पेंशन है उसमें से 4 माइक्रोलीटर वॉल्यूम आप 96 वेल प्लेट में 1 वेल में डालोगे तो उसका अप्रोक्सीमेटली सेल्स हो गए 10,000. ओके. ऐसे 4 माइक्रोलीटर आपको सेल सस्पेंशन डालना है और बाकी का 96 माइक्रोलीटर कंप्लीट मीडिया डालना है, और उसको इंक्यूबेटर में रखना है फॉर ग्रोइंग. तो जब आप डालोगे तो वो सेल्स मीडिया के साथ फ्लोट होते होंगे, but after 24 hours you can get your cells are achieved the proper morphology and it is become adherent cells. So by these way you can check the live as well as dead cell. Now cell revival मतलब आपके पास -80 डिग्री में सेल्स है तो उसको रिवाइव कैसे करते हैं. तो फॉर स्टोरिंग ऑफ द सेल्स वी आर यूजिंग क्रायोवायल. So you have to remove the cryovial from liquid nitrogen container or -80 deep freezer, or immediately you have to place the cells into 37 degree in water bath. तो जैसे आप वाटर बाथ में रखोगे उसका टेम्परेचर सेट करना है 37 डिग्री. तो ice melt and you are getting the cell suspensions. Then properly wipe the cell suspensions, cryovial outer side with IPA. Then ये जो भी cells suspension 1 ml है you have to transfer into the 15 ml tube, and in this 15 ml tube you have to add 9 ml of pre-warm media drop wise. through the wall of the tubes, mix gently and usko centrifuge karna hai 200 G for 10 minutes in cooling centrifuge. Later on, once centrifugation has been done, remove all the media and you are getting cell pellet, which is just another end of the tubes. Add 3 ml of complete media again with micropipette. You just mix it well. And वो जो 3 ml media with cell मिला, you just transfer into the T25 flask, add 2 ml more media, and incubate these flasks in CO2 incubator. So generally, you can get the cell growth within 8 hours, but after 24 hours, you can see that majority of the cells are adherent. Some cells are floating. So I think it is advisable to remove these floating cells and change the media. So whenever you are changing the media, every time you need to use 3 to 5 ml of complete media for cell growth. तो जनरली जब आप मीडिया चेंज करते हो तो इट्स कलर्स आर जनरली पिंकिश कलर बिकॉज इट हैविंग ए फिनॉल रेड बट once you once you check the cells in incubator and if your media color is yellow, that means there is a change in pH. Immediately you need to remove all the media, wash with PBS buffer and supplemented with the fresh media. So that is required, otherwise you will lose your cells. Now, what is the procedure for cryopreservation and subculturing? Okay, so suppose you procure the cell lines from the NCCS Pune, and once it achieve 90% of confluently, so you remove all the media and store the media in freeze. Okay, so why storage is required? Because sometimes what happen, whatever the media NCCS people are using and what media you are using, so companies are different. So sometimes, according to our observations, cells are not growing well. So what we are doing, we are preserving those media and filter with 0.22 micron filter. And if cells are not growing well, we are supplemented with NCCS media. So automatically cells are growing. So that's why preserving NCCS media that is required. So you have to take 90% of confluent cells, then remove the media and wash with phosphate buffer. उसके बाद you need to add trypsin EDTA solutions 1 ml. Then incubate the flask in CO2 incubator. Then add 2 ml of complete media to inactivate the enzyme. This is again important step. अगर आप 1 ml trypsin ले रहे हो तो वो जो एंजाइम है उसको इनएक्टिवेट करना जरूरी है, अदरवाइज इट इज ऑल्सो डिस्टर्ब योर सेल मोर्फोलॉजी. So how to neutralize it? So generally we are using double quantity of the trypsin, means you are using 1 ml of trypsins. You need to neutralize with 2 ml of complete media to inactivate the enzyme. Then mix it well. Then 1.5 ml, 1.5 ml दो append drop tube में निकालना है. Then you need to centrifuge it for a period of 10 minutes. And after centrifugation, remove the supernatant without disturbance of the pellets and then add. सपोज आपको क्रायो प्रिजर्व करना है तो 1 ml क्रायो प्रिजर्व मीडिया जैसा हमने आगे देखा वो स्लोली ऐड करके उसको मिक्स करना है. उसके बाद सेल को भी हम शॉक नहीं दे सकते. तो जैसे आपने क्रायो प्रिजर्व मीडिया ऐड किया तो डायरेक्टली यू कैन नॉट स्टोर एट -80 degree. So initially for a period of 1 hour, you need to store the cell in 4 degree, then transfer the cell in -20 degree. For that, you can use 1 degree cooler. And after -20 degree for 1 hour, you just store the cell in -80 degree in liquid nitrogens as well as deep freezers for a period of 2 to 3 months. After that, you need to revive it once to check whether they are growing or not. So this is a procedure for cryopreservation and subculturing. सबकल्चरिंग के बाद सपोज आपको ऐसे में यूज करना है तो जैसे मैंने बताया कि अपन ने 2 वायल लिया 1.5 ml का, तो उसमें से 1 वायल में पीबीएस डाल के उसको सेंट्रीफ्यूज करना है और उसमें से पीबीएस सॉल्यूशन में जो सेल्स है उसको एक काउंटिंग में लेना है. तो 1 ml विथ पीबीएस cell suspension, and 1 ml trypan blue generally we are using for cell counting. Okay. Now, generally we are checking the cytotoxic initial preliminary anti-cancer effect by using MTT dye. So there are various assays are there, MTT assay, SRB assay. But generally we are using MTT dye because which is well-established procedure and well-established assay. So MTT basically it is a kind of chemical kind of dye. So how they are acting. So MTT dye, which is react with mitochondrial reductase enzyme, which is present in the cells, and whatever the tetrazolium ring are there, so that is converted into the formazine complex. Okay, so this is tetrazolium dye, we can say, and once it react with mitochondrial reductase enzyme, it is converted into the formazine complex. Okay, so how to check whether cells are kills or cells are live? So when MTT dye react with mitochondrial reductase enzymes, so they are forming a formazine complex. Okay. So once your cells are live, that means mitochondrial reductase enzymes are also there. So they are react with MTT and they are producing more intense purple color. And sometimes if cells are killed, so no mitochondrial reductase enzymes are there, so they are not react with MTT dye and they are not form a formazine complex. So by checking the absorbance in ELISA plate reader at 570 nanometers, you can get the exact idea of the cytotoxicity or cytotoxic effect of your chemicals. So basically it is a 3 days assay. So in first day, what we are doing, first day we are seeding the cells, and generally we are seeding 10,000 cells in each well and keep the plate in incubator for a period of 24 hours in CO2 incubator. Next day we are observing the cell whether they are attaching or not. तो ये जो 96 वेल प्लेट है उसमें जो वेल है वो सारी वेल में सेल्स पूरे ग्रो नहीं हो जाते तब तक अपन ड्रग ट्रीटमेंट नहीं दे सकते. So once you 24 hours generally majority of the cells are growing well, but some cells are there, they are slowly growing. Its doubling time is less. So in such cases, we need to keep the plate in incubator for more 24 hours. So day one generally we are keeping for 24 hours. If cells are not growing well, you keep for more 24 hours. Next day, it is called drug treatment. So generally, drug treatment is for 24 hours, but certain formulation studies, they are checking for 48 and 72 hours. And last day is MTTS. Okay. So drug treatment कैसे देते हैं? तो based on different concentrations, we need to prepare a stock solutions. Okay. So suppose you are knowing the molecular weight of your compound, then you can calculate in micromolar. So for that, various molarity calculators are there. Sigma molarity calculator is there. You just enter the molecular weight of the drug, how much volume you require. So generally 1 ml, 2 ml volume is sufficient. And what is the concentration? So generally we are using 10 millimolar concentrations. Okay. So अगर आपको मोलिकुलर वेट पता है सो यू नीड टू ट्रीटेड विथ माइक्रोमोलर कंसंट्रेशन. अगर कोई फॉर्मुलेशन से कोई प्लांट एक्सट्रैक्ट है तो उसका अपन माइक्रोग्राम पर एमएल स्टॉक सॉल्यूशन बनाएंगे. ओके. देन इनिशियली वी हैव टू प्रिपेयर 10 मिलीमोलर सॉल्यूशन. सो उसमें से 100 लेना है, 900 ml फ्रेश मीडिया यूज करना है. सो इट इज कन्वर्टेड इनटू द 1 मिलीमोलर इक्विवेलेंट टू 1000 माइक्रोमोलर. एंड उसके बाद N1V1 का फॉर्मूला अपन लगाएंगे. ये आपका स्टॉक सॉल्यूशन हो गया. उसमें से आपको कितना लेना है, ये अपना डिजायर कंसंट्रेशन, मतलब उसको सेल को 10 माइक्रोमोलर पे ट्रीट करना है और 300 माइक्रोलीटर जो 96 वेल प्लेट होता है उसका कैपेसिटी है. तो 3000 डिवाइडेड बाय 1000 अपन करेंगे तो 3 माइक्रोलीटर, मतलब जो स्टॉक सॉल्यूशन है उसमें से 3 माइक्रोलीटर आप ड्रग लोगे और बाकी का 297 अपन ट्रेस मीडिया डालेंगे, तो वो वेल में कंसंट्रेशन, ड्रग का कंसंट्रेशन होगा 10 माइक्रोमोलर। 20 लेना है तो 6 लेना पड़ेगा, 40 लेना है तो 12 लेना पड़ेगा, ऐसे उसका कैलकुलेशन करते हैं। फिर फर्स्ट डे होता है कि सेल को ग्रोथ करना है, सेकेंड डे इट इज ए ट्रीटमेंट फेज, ट्रीटमेंट हो गया, नेक्स्ट डे आफ्टर 24 आवर्स ऑफ द ट्रीटमेंट you need to remove the 96 well plate from the incubator and check the morphology in inverted microscope, and you also preserve the pictures for that. Okay, because sometimes in thesis you need to show the pictures how morphology is disturbed. Okay, and then after you remove all the media from all the well, then add 100 microliter of DMSO solutions. Sorry, after drug treatment on third day, first we have to add 25 microliter MTT dye solution in each well. Then keep the plate in incubator for a period of 2 to 3 hours. Later on, अगर सपोज सेल लाइव है तो इट इज रिएक्ट विद एमटीटी डाई एंड वो फॉर्मोजेन कॉम्प्लेक्स फॉर्म करेगा। अगर सेल किल हो गए तो वहां पे कोई कलर डेवलप होगा नहीं, क्योंकि फॉर्मोजेन कॉम्प्लेक्स फॉर्म नहीं होगा। So after 2 to 3 hours of MTT dye treatment, then you remove the plate from the incubator, remove all the media from the pipette, and then add 100 microliter of DMSO solution. तो यहाँ पे DMSO का रोल क्या है? So DMSO, it is only solvent which is dissolve the formation complex. Okay, तो जो भी फॉर्मोजेन कॉम्प्लेक्स सेल के साथ फॉर्म हुआ है एमटीटी की डाई के साथ रिएक्ट होके, उसको डिसॉल्व करने के लिए वी आर यूजिंग डीएमएसओ। ओके, तो डीएमएसओ डालना है, देन कीप द प्लेट इन इंक्यूबेटर फॉर अ पीरियड ऑफ 30 मिनट एंड रिमूव द प्लेट, मेजर द रीडिंग एट 570 नैनोमीटर। तो 570 नैनोमीटर क्यों रखते हैं? बिकॉज lambda max of MTT dye is 570 nanometers. Okay, so sometimes I have observed the variation in absorbance. Sometimes we are getting wrong absorbance, so it is due to the its wavelength maxima. So how to check? So you need to prepare MTT solutions and keep the MTT solution in UV, and then check the lambda max. If it is 540 nanometer you are getting for your dye, so you need to check absorbance as 540 nanometer. But in majority of cases, you are getting the 570 nanometers. And you have to take two reading. One reading is at 0 hours, another reading is 24 hours. And you can get exact difference between these two reading, because formation complex takes time to dissolve. So सपोज आपने 0 आवर्स के बाद रीडिंग लिया तो काफी सारे सेल में लाइट कलर होगा। आफ्टर 24 आवर्स इट मे बी कन्वर्टेड इनटू द इंटेंस पर्पल कलर इफ सेल्स आर लाइव। So this is MTT assay. It is a cytotoxicity assay. We are getting preliminary cytotoxic effect through MTT assay, and through this assay we can calculate the IC50 value, minimum inhibitory concentration to kill 50% of the cells. और ये जो IC50 वैल्यूज आई वो आप फर्दर एंटी कैंसर स्क्रीनिंग ऐसे के लिए यूज कर सकते हो, लाइक डापी ऐसे करना है, वुंड हीलिंग और स्क्रैच ऐसे करना है, या फिर सेल साइकिल एनालिसिस करना है। इन ऑल दीज ऐसे यू कैन यूज IC50 वैल्यू। ओके, तो MTT is a preliminary assay to check the IC50 value on which concentrations your drugs are acting. Then और एक ऐसे आता है that is called reactive oxygen species. Okay, so generally these species are highly reactive oxygen containing molecules, and generally they are generated as a byproduct of normal cellular metabolisms. So generally they are formed as a superoxide anion, hydrogen peroxides, hydroxyl radical, singlet oxygens. So these all are the called reactive oxygen species which is generated by byproduct of the normal cellular metabolism, and these are the sources of ROS in cells. So it is mitochondrial electron transport chain, NADPH oxidase enzyme which is called NOx enzyme, peroxisomes, certain inflammatory responses which is responsible for ROS generations, and if exposures of the radiations, drugs and xenobiotic. So if you are expose the chemotherapeutic agents, expose the drugs, expose the radiations, so they are generate the ROS in the cells. So these are the sources. Now, why ROS are studied in cancer drug screening? So generally cancer cells are exhibit elevated basal ROS level compared to normal cells. And anti-cancer agents often increase the ROS beyond its cellular tolerance limit, and it disturbs the redox homeostasis. That means it imbalance the oxidation reduction kind of reactions. So that's why it is required, and it is a key mechanism. ROS generation, it is a key mechanism of many chemotherapeutic as well as the natural compounds, and it is helps to check whether it is a ROS mediated cytotoxicity or non-oxidative mechanism of cell death. So these are the actual pictures. So what are the procedure for ROS assay? So cells are seeded in each well plate. So in 96 well plate, whatever the wells you require, you just seed with the cells. Then after 24 hours, you need to treat the cells with the drug. So here we are using IC50 value for the treatment. Okay. So once it is treated, on next day in MTT assay, we are using MTT dye. In ROS, we are using DCFH DA dye. Okay. It is a basically fluorescence dye, and incubate 96 well plate in incubator for a period of 30 minutes. And that is react. If ROS generated, then generated ROS react with this dye and which produce the DCF. Okay, and whatever the DCF are produced, they are measured by using we have a Perkin Elmer Victor Neo multimode plate readers. So from these plate reader you can observe it. Now what observations you are getting? So this is control means untreated. So where you can see that there is no fluorescence are observed. These are the treated who treated well, where we are treated these well with our anti-cancer drugs. So where you can see this kind of fluorescent spotting. So that means once you treated your drug, which react with the cells at cellular level, and they are producing the ROS level, and produce ROS react with this DCFH DA dye, and which convert this dye to fluorescence DCF, and that gives the fluorescence. Okay. So through that, you can conclude that through ROS assay, your drugs are working on particular cancerous cells. They are producing the ROS level compared to its normal limit. And through this dye, we can check whether what kind of morphological image we are getting in control and what kind of morphological image we are getting in treated cells. Now next is wound healing as well as scratch assay. Okay. So why this assay is required? So in scratch assay or in wound healing, what we are doing, once cells are growing, and then we are keeping one scratch in 96 well plate. And if it is control, means treatment is not given, so cancer cell having a migration property. Okay, so data are migrated, and whatever the scratch we are observing, they are sorted. And in case of we are giving anti-cancer treatment to these kind of cells, and if we mark a scratch in the treated cells, they are lost their migratory property, and migration is not possible. Okay, so cancer cell metastasis depends on the cell migrations, where tumor cell migrate from primary site to invade the surrounding tissues and form metastases that we know. Now, many anti-cancer drugs are designed to inhibit this migration, not just kill the cells, but also inhibit their movement. And these wound healing assay help to identify the anti-migratory as well as anti-metastatic potential of these compound. So sometimes you are using anti-metastatic effect. So for that, you have to perform this kind of assay. And generally it is performed for solid tumors like breast, lung, colon, glioma, and melanoma. Now, what are the principles? So, a uniform scratch or wound you have to produce on confluent monolayer cancer cells. Then you have to treat the cells with test drug, your compound. Suppose if you want to compare with the positive control, so in case that you have to use known migration inhibitors and vehicle control means untreated. So generally, you have to observe or monitor the migrations of the cells in wound area for a period of 0 to 48 hours and reduce the wound closure, that means inhibition of cell migration. So how we are getting the results and what we observe, I'll show you. Now, what kind of informations we are getting from these kind of assays? So first informations we are getting that effect on cell cancer migrations. Whether cancer cells, once it is treated with anti-cancer drug, they are migrated or not. We can check their collective lateral movement of the cells and whether they are indicated with that your compound inhibit the migrations or delay in wound closures or completely block the movements. Okay. So for delay in wound closure, that means you have to observe the cells for a period of 48 to 72 hours. Next is you can also check the incomplete wound closure, which suggests that suppression of the metastatic behavior as well as interference with cycloskeletal dynamic as well as focal adhesion as well as various signaling pathways are also involved. So in mechanistic insights, when you combine with other assays, so these assays directly reflect the effect on actin cytoskeleton remodeling as well as cell-cell or cell matrix interactions or certain migration-related pathway such as PI3K, KT, MAPK, ERK, FAK, SRC, and EMT. You can also get dose-dependent responses in this study, which helps to identify minimum concentration required to inhibit the migrations. And you can also get the idea about the concentration ranges that affect the migration without causing the cytotoxicity. Now, migration studies should be ideally be performed at sub-cytotoxic concentrations. So, as I said, IC50 value which are getting from MTT, XTT or reserve urine acid, these all are the cytotoxic acid. And we can also get the idea about time dependent effect means you have to compare the wound closure at different time interval. So from that you can get an idea at 8 hours we are getting or migration of cells are stops. In some cases we are getting 24 hours migrations of the cells has been stopped. So these are the results. So this is control and this is treated. Okay. At zero level, you just here you made a scratch. So here there is no cell has been observed. Okay. But after 24 hours, as I said, cancer cell having a migration property. So they are migrating well and these distance are shortened in control that you can observe through microscope. But in treated, here you can see same distance you are getting. That means your compounds are acting as an anti-metastatic agent, anti-migratory property. Well, cells are not migrated and distance are not shortened. So these results shows that your compound having a anti-metastatic effect. Okay. So how to perform these assays? So same way 96 well plate you have to taken, you have to seed the cells. Then cells were treated at IC50 value, which is known after MTT assay. After confluency, a wound was created. Wound means you have to make a mark with tip of the micropipette and at different time interval you observe the cells under inverted microscope. Next is cell cycle analysis. Okay, so what we get from cell cycle analysis? So from cell cycle analysis, we have an idea that whether your tested compound is acting on which cancerous cell stage. Okay, so we know cancer cell cycle G0, G1 phase, S phase, G2 phase, and last is mitotic phase. Okay, so if your compound is acting on G0, which is called sub G1 phase, or G1 phase, then it is good compound because which is initially stop at growth phase. So that gives a better anti-cancer effect. So why we perform cell cycle analysis to evaluate the effect of anti-cancer drug on specific cell cycle stage to determine whether drug induced cell cycle arrest at which specific phase, whether your drug is acting on G0, G1 phase, S phase, G2M phase. You can also understand the mechanism of actions of your candidates. You can also distinguish the difference between cytostatic means growth inhibitory and cytotoxic means cell killing activities to identify the drug that sensitize the cell to apoptosis by disturbing the normal cell cycle checkpoints. And through that, we can also get an idea to structure activity relationship of lead compounds and to complement cytotoxic assay by providing a mechanistic insights. Now, first informations we are getting from cell cycle analysis is distribution of cell in different phase. So generally 3 phase we are getting idea about 3 phase we are getting G0, G1 phase. We know that it is a cell growth and preparations of the DNA synthesis phase. S phase where DNA replication is taking place in cancerous cells, and G2 phase, which is called growth 2, and N is mitotic phase. So which is prepare the cells and executions for the mitosis. So we get idea about different phase arrest, whether your drug is acting on G1 phase or G2 phase. Then next, we can get an idea about phase specific cell cycle arrest. Suppose your drug is acting on G0, G1 arrest, so that inhibit the cyclin D or CDK4/6 activity. So cyclin D and CDK4/6 generally it refer to a regulatory proteins complexes that control the progressions of the cell cycle from G1 phase to S phase, where cyclin D bind to the CDK4/6 and promote the early G1 progressions by phosphorylating RB proteins. And cyclin E generally bind to the CDK2 proteins and drive the G1S transitions and initiations of the DNA replications. S phase arrest means which interfere with DNA synthesis and replications. And G2M arrest means it disturb the microtubules or DNA damage. So these are the phase specific cell cycle arrests. Sub-G1 population that indicate the DNA fragmentations and apoptotic cell death is occur, which suggests the late stage apoptosis induced by the drugs. Certain checkpoint activations we should know. Say for example, G1S checkpoint means there is an involvement of the certain gene like P53, P21 for inductions of the cancers. G2M checkpoint means ATM, ATR mediated DNA damage responses. And drug selectivity we should know, as well as dose and time dependent effect we should know. This is how we perform the cell cycle analysis. So for cell cycle analysis, we need to seed the cell in T25 flask. So generally for seeding the T25 flask, we need to require 1 million cells. Then treated the cells where centrifuge and pellet were collected. So whatever the treated cells are there, we need to trypsinize it. After trypsinization, remove the trypsins or neutralize the trypsins by using complete media. Then again centrifuge it, remove the solutions, and whatever the pellets we are getting, it should be fixed at 70% ethanol. Then we should to add RNase A, which disturb or we degrade the RNA because we require the DNA. So whatever the RNA are there, it should be degraded before run the cell cycle analysis. Then propedium iodide dye should be added, and then run your sample in flow cytometry. So we don't have a facility of flow cytometry, but we just prepare the cells for flow cytometry, and we are performing these assay at GCRI, Gujarat Cancer Research Institute. So we are getting this kind of graph. This is control. This is at 10 microgram per ml. This is 50 microgram per ml and 100 microgram per ml. So how to read these results? So you just check it. If sub G1 8% inhibitions, sub G1 at 10 microliter 9%, sub G1 50 microgram per ml 50% and 100 microgram per ml 79%. तो आपकी ड्रग कौन से फेज कौन से स्टेज को अरेस्ट करती है, तो हियर वी कैन कंक्लूड दैट आवर ड्रग विच इज एक्टिंग ऑन सब G1 फेज. नाउ व्हाई नॉट G1S एंड G2M? तो यहाँ पे G1 60%, G2 पे 73. फिर जैसे कंसंट्रेशन बढ़ती है तो ये वैल्यूज कम हो रही है. दैट मीन्स वी आर नॉट कंसिडरिंग दीज गिवन एरर. सेम वे हियर 12%, उसके बाद 8%. सो वो वैल्यू कम हो रही है. So that's why we are not considering. Same way G2M 20% and here 10%. Again, these values are less. So which value we have to consider? So if percentage are continuously increasing, so 8, 9, 50 and 79%. So they are continuously increasing when drug concentrations are increasing. So we have to consider that our drug is acting at sub G1 phase. Next is DAPI assay. So DAPI is a dye. It is a fluorescence dye which is used to check apoptotic effect. Okay. So why DAPI assay is useful in anti-cancer drug screening? So first is inductions of apoptosis is a primary goal for anti-cancer therapy. So whatever the DAPI dye is there, which is bind strongly with 80 region of the DNA and allow the clear visualizations of nuclear morphology of the cells. If we treated your cells, cancerous cells, with anti-cancer drug, which often causes chromatin condensations and nuclear fragmentations, how it looks, we'll see in the results. DAPI staining also provide direct visual evidence of apoptotic cell death, and which is confirmatory assay alongside cytotoxicity and biochemical apoptosis assay. So what are the principles of the DAPI assay? So cells are treated with the test compound at selected concentrations. Then cells are fixed, usually paraformaldehyde as well as the methanol. Then it should be treated with the DAPI. So DAPI will penetrate into the cells and bind with nuclear DNA. If your drug is anti-cancer drug having anti-cancer potential, so which damage your cells, which damage the DNA and it's change the morphologies. So whatever the nuclei are visualized under fluorescence, so it is emitting blue fluorescence, and change in nuclear morphology, which should be compared with the untreated control cells. If your drug having a proper anti-cancer property, so it induces the apoptosis and it gives the effect through DAPI staining. Now, what DAPI staining help? So DAPI staining generally help to identify normal nuclei. It should be, if you check the normal nuclei, it should be uniform, round. Just a minute. हॅलो
नाही दिलेच नाही आपल्याला केव्हा केव्हा घ्यायची पण ते एक नवीन
हॅलो
Sir,
Sorry.
yes.
DAPI staining generally help to identify normal nuclei, what your normal nuclei looks. It should be uniform, round, and light blue fluorescence. I'll show you the result. If there is early apoptosis, late apoptosis, as well as necrosis, means complete cell death, that we can get evidence of apoptosis inductions, means programmed cell death, we can get from the PSA. Then dose-dependent apoptotic response we can get, time-dependent apoptosis we can get an idea, and sometimes we can also check qualitative as well as the quantitative analysis. So in our laboratory, we are only performing the qualitative DAPI assay. So this is untreated cancerous cell where you can see each and every cells are in good conditions. They are well fluorescent. And these are our formulation, anti-cancer formulations, treated cell, where you can see three different kind of changes. These cells you can see it is in more or larger cell. So this effect is called cell shrinkages is taking place, means there is accumulations of the fluids are taking place. Then membrane labeling means certain apoptotic body or certain cells material which is going to start from the cells, which is called membrane labeling effect. And last, you can see small apoptotic body you can observe here. That means your DNA is totally damaged, cell DNA is totally damaged, and you are getting these three kind of effect. So this is qualitative assay. You can show this kind of effect. These are the other cell-based assay for formulation, cytotoxic. Cell viability assay is nothing but MTT assay. Proliferation assay, again, we are using MTT for that. Apoptosis, we are using DAPI. Cell cycle analysis, we are just discussing. Then gene expression analysis, you can do under cell culture technique. Protein expression analysis and cellular morphology assessment you can do. This is one of the study we have performed in Vero cells. Vero is a monkey kidney cells. So this is image A. Just a minute. Thank you. This is image A. These are the Vero cells. You can say cells are in good conditions. This is image B, where we are treated our cells with formulation. This is image C, where we are treated with placebo, and this is pure drug. Okay. So we communicated one paper. We have performed our formulations on cancerous cell lines, but reviewer asked to check whether at what concentrations you are getting effect on normal cells, whether your formulation which is affecting normal cells or not. So this is control, means we have not treated, this is treated with formulations, this is treated with placebo, and this is treated with drug. So from these results, we can conclude that there is no significant effect has been observed at placebo. Placebo treatment means whatever other excipients we are using, they are safe, we can say. But whatever the drugs are there, so drug having a certain effect on Vero cells. So we are getting this effect at 120 microgram per ml. So it is at higher concentrations. So we need to reduce the concentration. This is one effect on Vero cell line. So sometimes whenever we are communicating the paper to the journal, so they are also asked for these study. Check your formulation, check your compound, whether they are acting on normal cells or not. So we have performed this kind of study on Vero cells as well as NRK cells. This is neated NA formulations. We have check on lung cancer cell line. So this is untreated cell. You can see how it is well organized in their morphology. 5 microgram per ml, you can see that cells are not intact and you are observing the space and you are observing this kind of rounding of the space. That means there is some effect which is initiated at cellular level. 20 microgram per ml, you can check morphology of the cells are totally disturbed. And 40 onwards, you can see this kind of images, morphological images. That means your formulations, it is start acting at 5 microgram per ml and start gives activity which is 20 microgram per ml. Okay. So this is one of the studies. Another study is on LANCAP prostate cancer cell lines. So this is untreated cells. This is at 5 microgram. There is no effect. Here you can see 20 microgram per ml effect has been started. Cells are uh, reducing. And at 80 microgram per ml, you can see morphology of each and every cell has been changed. And 160 and 320, we are getting less amount of the cells. Even morphology is not there. So this technique generally it is based model system for cell biology interactions between disease causing agents and cell. We can check effect of drugs on the cells processing and toxicity testing we are using. Even virology, we are using this kind of technique. Whatever the COVID vaccines has been prepared, that is again based on cell culture technique and certain cancer research we are using. Genetic engineering, gene therapy, tissue culture, we are using this technique. So, participants, these are the outcome of our laboratory. So, these many articles we have published in 2025 with elsewhere Springer Nature, Taylor Francis, RSA Advanced, and ACS. These all are our collaborators. First one is Dr. Gayatri. Currently, is working in one of the pharma company at Canada. Is Dr. Ria. Is Dr. Gayatri's PhD student. Dr. Pinkal Patel, basically, he is a medicinal chemist working at Parole University, and she is our one of the collaborator. Dr. Swayam Prakash, basically working at our university, and his core area is artificial intelligence in drug discovery. So he has developed one AI-based docking tool. Just you insert the PDB ID, automatically this AI model gives you a number of novel compound which you need to synthesize. And they have also merged this tool with Autodoc software, and automatically you are getting the docking scores. Still, it is under validation stage. Dr. Suraj Mali is assistant professor at DY Patil University. Basically, he is a core chemist. He helped us in computational chemistry. Rahul sir from your college, he has a great computational chemistry background, so help us in computational study. Dr. Tesir is there. He is working at Amgen Laboratory, Hyderabad. He is also computational chemist. Dr. Vicky Jain currently he is pursuing his project at Belgium. He is also a chemist where I checking their compounds anti-cancer potency. Dr. Ashish, Dr. Mehul, they are from our, but Dr. Ashish just left our university and he is working at Parul University. They both are chemists and I'm assessing their compounds. Dr. Vishnu from Nasik, again, he's a good chemist. He has synthesizing compounds and tested their compound in our laboratory. This is whole our team. Students are working. Aarti is there. Dr. Roma is there. Adrija, Devraj from Bengal. Himisa. So they all are pursuing PhD. And these are the pharmacologists. They are helping us in laboratory. So with this, here I conclude. If anybody having any questions, I'll try to solve. Basically, I am a chemist, but during my PhD, I need cell culture technique. And in our university, we have a laboratory in 2012, but nobody knows how to use. So I have started study in 2015, and then initially we have collaborated with people. We are getting the samples, we are getting the projects, and right now laboratories are well working. So again, I am thank you to all organizer for inviting me to share my views on cell culture technique for anti-cancer screening. Again, from participants, if anybody require my help, I am always open for collaborations. Thank you. Thank you very much.
Thank you very much, sir, for such a informative and enriching session. Before we proceed to the vote of thanks, I would like to open floor for brief question answer session. Participants are kindly invited to ask their questions related to today's session.
Hello, sir. I have posted some questions in chat. Don't know whether these questions are feasible to answer or not. I don't have much idea. How do we can explore whether the sample molecule showing activity nanomolar range or macromolar range?
Okay. So, sir, what we are doing, if compound is not known, so we have started the concentration, which is started from 1 micromolar to 160 first time. So, if we are getting the result at 1 micromolar, then we are check those sample at nanomolar range. So, through that, we have an idea whether our compound is showing micromolar or nanomolar activities.
And
Next question is how do we can validate the absorbance correlate with actual cell numbers? So sir, जैसा मैंने principal में बताया कि MTT, how MTT works. So if cells are live, that means it react with MTT dye and it gives intense color. So its absorbance are more. If our drug having a effect, that means cells are not viable. That means cells are killed. So they are not reacting with MTT, and we are getting no color or a certain light color. So through absorbance, we can correlate whether cells are there or not. Is there any specific statistical method for IC50? So, sir, for IC50, I am using Prism software, but last few years I have observed that we are getting wrong IC50. So I have checked one paper. So through paper we have measured the IC50, and nowadays we are getting proper IC50. I will share this paper to you. Next is, does the reduced MTT signal correlate with pathway dysregulation shows in IC50 or histopath molecular data? So through MTT, we cannot check whether any signaling pathway is dysregulated or not. We are only getting an idea about the cytotoxic effect of the drug.
Okay, thank you, sir. I have one more question. So, is there any correlation between MTT response and the cell cycle arrest? Shall we establish any correlation?
Yes. So for whenever we are performing cell cycle analysis, so generally the concentration we fix. So we are getting IC50 value, which is getting from the MTT assay, and same IC50 we are using. And sometimes what happen we are getting +5 or plus minus of these IC50 value. So we are treated the cell on 3 different concentrations. So middle one is our IC50 value, and we are getting certain less concentration than IC50 and certain more concentration than IC50 value.
Okay, thank you so much, sir, for informative and such an insightful session. Thank you from my end and from my college. Thank you.
One more question is there, sir. Dr. Prashant is asking, how does sample preparations affect the cell cycle accuracy? So it is again good questions, because sometimes actually we have prepared sample for many times and we are not getting the results. So if your cells are not properly fixed, it is not properly run in cell cycle or we can say flow cytometer. So it is again good questions, and where expertise is required for that. How to distinguish between cell cycle arrest and cytotoxicity in result? So cell cycle arrest, we are getting that at which stage our drugs are acting. And cytotoxicity assay, that means it is completely killing of the cells. So at which concentrations it kills, and same concentration we are using in cell cycle analysis. So through that cell cycle analysis, we can have an idea that on cancer, which phase in cancer cycles, which phase our compounds are acting. I hope I answer all the questions. Thank you to all participants for good comments, and I'm also learning this technique in depth, because this is not my core area, but PhD ने सिखाया कि we need to learn other technique also. Thank you very much.
Thank you, sir. I now request all participants to kindly turn on your video to capture the attendance. Thank you. All participants are now requested to stay connected next session with stars soon.
So again, thank you very much to organizing team for inviting me and give me a chance to explore our laboratory's output. Again, thank you very much. If any participants having any questions, you can get my number from organizers. You can contact me. Thank you. I am leaving this Zoom meeting. Thank you.
Thank you, sir. त्यांनी म्हटलं मी लेफ्ट करतो.
The science of today is the technology of tomorrow. Good afternoon and a very warm welcome to all. Respected resource person, honorable dignitaries, respected chief guests, and respected faculty members, participants, in my proud privilege to welcome you all to second session of day 4 of the faculty development program on artificial intelligence as a catalyst for excellence in pharmaceutical education, research and innovation. And I am Ms. Ishwari Pasgare, Assistant Professor from Department of Pharmaceutical Chemistry at Dr. Rajendra Gode Institute of Pharmacy, Amravati. And I feel honored to coordinate this session. It is truly a privilege to bring together such distinguished experts and enthusiastic participants to explore the transformative role of artificial intelligence in advancing pharmaceutical education, research, and innovation. On behalf of the organizing committee and our institution, I once again extend a cordial welcome to all the participants. Artificial intelligence is the rapidly reshaping pharmaceutical science by enabling the faster drug discovery, accurate prediction, model, and data-driven decision. One of the most impactful sessions lies in AI-based ADME prediction, which significantly reduces time, cost, and failure rates in drug development. Today's session on artificial intelligence-based ADME in drug discovery is designed to provide deep insights into how artificial intelligence is revolutionizing early-stage drug development and molecular screening. It is now my privilege to introduce our esteemed resource person, Dr. Shashikant V. Bhandari sir, Professor at All Indian Sri Shivaji Memorial Society, College of Pharmacy, Pune. He has over 23 years of teaching experience, and 18 years of distinguished research experience. He holds M.Pharm and PhD degrees, and he successfully guided 7 PhD scholars and 56 postgraduate students. His course expertise includes drug design and development using advanced in silico molecular modeling techniques such as QSAR molecular modeling techniques, ADME prediction, DFT studies, and molecular dynamic stimulation across the therapeutic areas, including anti-cancer, antiviral, antibacterial, and anti-HIV, anti-inflammatory, anti-tubercular, and anti-convulsant agents. With over 73 international publications, an H-index of 15, and more than 1,200 citations. He has made remarkable contribution to the pharmaceutical research. He has also been the recipient of numerous prestigious research grants and national awards, including the Best Research Mentor Award, this ISMTM in 2021, and Outstanding Publication Award in Molecular Modeling. His deep expertise in AI-based computational drug design and ADME modeling makes him the perfect speaker for today's topic. On behalf of the organizing committee, I respectfully invite Dr. Shashikant Bhandari sir to kindly take over the session and enlighten us with his valuable insights on artificial intelligence-based ADME in drug discovery. Hello, sir.
Yes, thank you very much. Am I audible?
Yes, sir.
Yeah, thank you for a very good introduction. I'm really privileged to hear all about myself. One thing only I would like to correct is in total I have 26 years of academic experience. You might be correct, 23 years of research experience. I'm really delighted to see the kind of huge number of participants all across, I suppose, Maharashtra and India. So first of all, congratulations to whole organizing team for having invited so many participants, and that too continuously involved. I was attending previous session. I could see hardly few members left the meeting because in general the virtual meetings the attendance keeps on moving on and off. I'm really happy to see that all are it's a very serious crowd. Before wasting time, let me share my screen. First of all, I would like to thank Professor Rahul Javadkar sir and principal and whole organizing team for giving me opportunity to interact with all these maybe seasonal researchers and some are budding researchers who would really shape the future research and development in pharma profession. Before we begin with, I understand that everyone is really attending very seriously. So I would just roughly, in short, go through some important points. Why AI-based drug design and discovery? The theme of the program has already informed you about the importance of this particular topic. So I will not waste much of the time. These are all my contact details. Anybody can get in touch. Of course, organizers added me in the group. Why molecular modeling? Just to reduce cost, time, energy, efforts, and to improve accuracy of research and development. So right now, till now, we used to use molecular modeling tools like QSAR, toxicity prediction, absorption, distribution, metabolism, excretion, toxicity prediction. But now, with the advent of AI, we have developed our own indigenous tools. The inventors are from Pune itself, Maharashtrian, and I'm really happy to say that we could develop a product which is in actually a comparable with the very well known and famous product like Rajendra, molecular orbital environment, mall soft and all. We would definitely witness the outcome of the research work as we keep on going ahead. Before that, I would
Yeah. Is there any problem with audio?
No, sir, we
Hope
can.
not. Before that, I would just thank you very much, sir. I would just request everyone to kindly you know
Hello, sir, your mic is off.
is it okay now?
Yes, sir, it's okay.
Yeah. So I have given link for registration yesterday evening. Most of you have already registered. I would just like to know how many of you have registered. You can show from, so that will really motivate me and keep me inspired throughout the session. That these are all interested participants. So these are some successful stories or examples of the drugs invented by artificial intelligence. You can take screenshots. I really don't want to invest much time in giving examples of the success stories others have invented. I would love to see some of you ending up with some discovery, maybe within a span of 6 years or 1 year. But through AI tool, it takes a very less time. In under 12 months, people have succeeded in ensuring that the product reaches to clinical trials, up to phase 3, phase 4. So it's so short span of time. Otherwise, we have seen and heard that drug discovery requires at least 8 to 10 years, more than $200 billion of investment, a lot of manpower, scientists working from different faculties. So AI has given some Sumotomo, developer pharma. It's a Japanese company inventing BSP 1181 for obsessive compulsory disorder. Next example is it required only less than 12 months to get a drug in the phase 3 or phase 4 clinical trials. Then in silico medicine, COVID-19 pandemic, within 2 months, people could ensure a drug for treatment of SARS-CoV-2. HIV protease inhibitors were invented using computational tools like saquinavir, imatinib, one of the most important anti-cancer agents used to treat different types of chronic myeloid leukemia. Then oseltamivir, drug used to treat
Hello, sir.
hepatitis.
हॅलो, सॉरी टू डिस्टर्ब यू. सर, ऐकू येते माझा आवाज?
Yes.
सर, तुमचा आवाज प्रॉपर येत नाहीये.
Okay.
हा,
Just a moment.
तुम्ही ब्लूटूथ इयरबड्सनी बोलताय का? त्यामुळे कदाचित आवाज नीट येत नाहीये.
Maybe.
हा, आता ट्राय करून बघा, मग आम्ही सांगतो तुम्हाला.
Yeah, मी ब्लूटूथ आता बंद केलेलं आहे.
हा,
Am I audible now?
आता व्यवस्थित येत आहे सर. आता कंटिन्यू करा. सॉरी सर, सॉरी टू डिस्टर्ब यू.
Fine. No. Thank you for letting me know.
Okay, sir.
In dorzolamide, a drug used to treat glaucoma. These are all successful stories or inventions using computational tools. Biologics and antibiotics were also invented by companies like AstraZeneca, Accelries. Now these guys have invented a lot in Schrodinger's molecular modeling tools, therapeutics, then Agios Pharmaceuticals, Takeda Pharmaceuticals, and these are all drug discovery tools. Random screening, non-random screening, drug metabolic studies, clinical observation, rational approaches to lead discovery. We would invest our time in getting understand lead optimization. What is the role of AI tools? Okay, so I will not repeat all these drug discovery tools. So these are all AI models and the tox cost data, 93 papers, toxicity endpoints, various AI tools available in the market, and what type of toxicities they are really predicting. So this is literature survey wherein 2 papers are reported for pulmonary toxicity, 3 papers reported neurotoxicity prediction, 4 papers reported cytotoxicity, and in 4 papers, developmental toxicities were reported. Now we have the tools which are accurate to the extent of 94% so far. It predicts more than 40 plus properties. It is used to expedite the predictions using de novo design, that is target specific novel structures. Then we have AI based or machine learning enhanced docking suit. Now these are simple steps. Simply upload the protein, select the grid, and then it automatically prepares the grid. You have to just enable the active site data, subject it to docking. You get the results in few minutes. You need not input lot of scripts, just like we need for some free tools available in the market, like Vina, Autodoc Vina, and all. So here we have improved the accuracy of AI detection and paraphrasing to 97%. Now I'll straight away take you to the advantages of AI tools. Here we have a G-Info suite. The prediction speed is seconds to minutes. Other methods take some days, weeks, and other platforms few hours. Accuracy is more than 90%. Multi-parameter analysis is very comprehensive details we get. We get integration capabilities. Cost efficiency is very high. So such kind of molecules can also be designed using our AI tools. These are some examples of recent analysis. Now I'll straight away show you demonstration of certain tools. I hope everyone has activated the So here we go. This is G-Enforce. Once you log in, you can go to tools. Are you all ready? I'm happy to see that number of participants are increased to almost more than 144, 143. So around 10, 12 participants are more. Let us start with tools. What we need to do is we'll start with toxicity prediction first, because this is applicable to all specializations in pharmacy, right from pharmaceutics to quality assurance to pharmacology to medicinal chemistry. So such kind of window is open. I hope the screen is visible, right? So here you can use these tools to draw the structures, or you can directly input the structure in the form of smiles. Smiles you can get anywhere on internet platforms. Say for example, I'll just write osimertinib and smiles, right? So here National Institute of Health, NIH, or PubChem database, you can just click on NIH site and then you'll be able to see all the details of osimertinib right from PubChem ID, structure, primary hazards, molecular formula, synonym, everything. Just go down till you get IUPAC name and to smile here. I'm just copying the smile, trying to make it as simple as possible. Then here we are going to ginfos.com, paste the smile, name the molecule osimertinib. For information of everyone, osimertinib is phase 4, or you can say 4th generation EGFR inhibitor. If I click on analyze toxicity, just count. It took few seconds to predict the toxicity. Here you get profile. It says molecular weight, total polar surface area, hydrogen bond acceptor, QED, log P, hydrogen bond donor, rotatable bonds. Taking help of all these properties, it has predicted the compound will have acute toxicity, and LD50 dose is 949 milligram per kg. It comes under class 4 toxic compound. What type of toxicity it is predicting? Endpoints are hepatotoxicity, it is inactive, showing in green, and inactive, it means it is non-toxic. Then carcinogenicity, mutagenicity, the compound penetrates blood-brain barrier. So it is showing active because it may show high CNS toxicity. Respiratory toxicity, neurotoxicity, immunotoxicity, cardiotoxicity, nephrotoxicity, ecotoxicity, clinical toxicity, developmental, endocrine, skin sensitization, and phospholipidiosis. So all these toxicity endpoints are predicted. If you want the report, you just click on download report. You will get a fantastic osimertinib tox analysis report downloaded to you. So you need not prepare. You get a tabular form, gene tox, advanced toxicity prediction. Here you will get a conclusion LD50, which class under which class it is classified as hepatotoxicity inactive, and the confidence level is given in the bracket 50%. 58% confidence it shows not carcinogenic. 35% confidence is not respiratory toxic. Blood-brain barrier 70% confidence, quite reliable. Okay, so overall risk level is moderately toxic, and here you get all these parameters and then details, endpoint, prediction, probability, and reliability. So very few tools available in the market would give you such kind of ready-made reports, ready to get incorporated in the literature or research paper. So that is all about toxicity. If you see, I'll immediately get the results in my email. Within few minutes, you get the results in your mailbox. Okay, let us wait for a few minutes. Every result is available online in your email box. Okay, maybe few minutes we'll get the details with us. Next one. Is there any doubt about prediction of toxicity? If not, we'll go to demonstration of another tool. Now we'll demonstrate prediction of ADME, right? What your new chemical entities you're designing, you can predict. Now here I am taking example of a marketed drug so that you can access the literature and compare what sort of predictions our tool is giving and what is reported in the market. So here straight away you can copy the smile of osimertinib from PubChem database, then paste here. I'll give you 5 minutes break for you to predict at least two properties, and then we can proceed to other tools. So please pay your attention right now with the demonstration part. Within 5-10 seconds, we are ready with the property. Here it is summary properties, polar sulfate, right from molecular weight, molecular formula, log P, hydrogen bond donors, hydrogen bond acceptor, polar surface area, rotatable bonds, QED that defines, gives you idea about drug likeliness score. And then you get idea about these are the, important screens used. You'll find all these parameters incorporated in one single AI tool very rarely. Here we are taking help of Lipinski's rule of 5, Gauss filter, Weber filter, Egan filter, Muger filter, and at the end you get bioavailability score. So it is violating one property of Lipinski's rule of 5. If you are belonging to pharmaceutical chemistry or medicinal chemistry, you can definitely make out which parameter it has crossed. Can anybody? This molecular weight If you want your compound to be drug-like, to show drug-like pharmacokinetic profile, its molecular weight should be less than 500, log P should be less than 5 hydrogen bond donors, 5 to 10 hydrogen bond acceptors. Again, 10 is the limit. Polar surface area, more the area, more would be its possible, water solubility. Rotatable bonds probably the count as exceeded, right? Many rotatable bonds increases molecular flexibility and that may reduce oral bioavailability. So we added the interpretation part itself here itself. Low drug likeliness score potentially problematic. This QED parameter. Now you can simply export this to PDF or export this file to Excel, wherein I'll just write osimertinib admit prediction. Let's see what parameters it is giving and how the report is user friendly. Here in Excel, you'll get all these properties and their values over here. Ready-made smile is incorporated. Lipophilicity, if you see how many parameters it has predicted, 13 under molecular properties, 9 under lipophilicity, 12 under water solubility, means 21, 34. Pharmacokinetics, 1, 2, 3, 4, 5, 6, 7, 8, 9, 43, and then 45, 47, 49, 52, 53, 4, 5, 6. Total 56 parameters are predicted. Again, these results would be emailed to you in the inbox. Here, G-Indoxid license details. If you see Gene Force, what your email ID or registered, you'll get all the details in your inbox. Now let us. So this is ADMET analysis report, right from structure to all these parameters. I have shown you the Excel sheet. You can take a print of the same also. So this is all about two properties we have predicted so far. Shall we go ahead to gene bio predictor, or you want to experience by predicting for your own molecule? Kindly advise me. You can write in WhatsApp group also. It is very easy because I don't think you really need much practice. You don't have to write any script. You do simply give input about structure. Structure input you can give. You can upload the structure in the form of SDF file, .mol file, .mol2 file, or directly smiles even can be given. Now let us see how gene biopredictor works. Again, toxicity. I want bioprediction. Okay. Here you can input any compound, any structure. Say for example, I'll take paclitaxel. We'll search its smiles. It is reported anti-cancer compound isolated from natural sources. So here we get the smiles of paclitaxel. It's a relatively bulky structure. You should not make any mistake in copying the smile, because giving wrong input will definitely give you wrong answer. So this is details of paclitaxel. Say for example, you are natural product expert or you are a medicinal chemist and you have isolated some naturally occurring active principle and you don't know where it will act, what would be its mode of action, what would be probable activity. Just give its input here. You'll get all the details. The structure is already on the screen. Click the predict part. It has predicted bioactivity score only 0.44, bioavailability score 39%. Now let us see what are different targets it has predicted. It says the compound is, it has 45% probability that it will inhibit monoamine oxidase B. And the confidence level limit is again good, more than 60%, that is more than 50%. 70% confidence, MAO-B inhibitor, dipeptidyl peptidase 4, probability is 44% and confidence limit is 61%. So these are all different properties it is taking into consideration while predicting its bioactivity: molecular weight, lipophilicity, H bond donor, acceptor, rotatable bonds, aromatic ratio, fraction of sp3 carbon atoms, and then it predicts 20 different targets. I would insist you to rely on the targets which are above 50% confidence limit. Okay. Now, if you want this report to be exported, just click on export. It will export as Word. I'll rename this bioactivity prediction for paclitaxel. Here you get prediction, score, confidence limit, bioavailability, key properties, and then it is giving rank. The first two are quite good. Trust on that. The rest you can ignore. The reference is given: analysis, interpretation, moderate, violates 2 Lipinski's rule, bioactivity prediction. It has low bioactivity. Significant modifications are required in order to make the compound very potent, effective, and drug-like. The primary target predictions are these two. Secondary target is this one. Then at the end of a report, it clearly says that it was generated using gene biopredictor machine learning models. Results are predictions and should be validated experimentally. That is all about bioactivity prediction and its example. Are you all clear? I'll have to check your messages. So far we have seen toxicity prediction, ADME prediction, gene bio prediction. Now this is something which is very interesting, but it will take time. Let us see. Here you can give PDB ID of the target and then it will design compound for you. It will generate novel molecules. Say for example, I will take something which is already used because the data is available over here. And if you say target search results, generate novel molecules, it generates some molecules. It is taking more time. I will show you the example of previously generated molecules. If I say view, I'll go to results. The target given was 41BB. Okay, this particular enzyme PDB code was given, and then it has given generated some novel molecules like this: molecule, molecular formula, molecular weight, log 3, Okay, hydrogen bond donors, acceptors, total polar surface area, rotatable bonds, novelty score, drug likes, likeliness, everything. So likewise, you can either see table view, say 10 molecules, and then novelty score is also given. Novelty score does not mean that the molecule is novel. Novelty score means that the molecule's novelty for its reports for that particular enzyme or target inhibition. Nobody has reported this molecule for inhibition of 1BB. So 1BB is the name of enzyme or protein. Okay. So novelty above 50% or 60% is always welcome. So all these parameters are predicted. In level 1, it can predict 100 molecules, level 2, 1000 molecules. Sorry, level 1 itself, 1000 molecules. It can predict 1000, 10,000. Again, it depends on, efficiency of your system. It should be at least Core i5 machine with maximum random access memory, RAM, very high, hard disk capacity. Processor agar अच्छा है तो you can get the results faster. So that is all about gene mole tool. As far as registration is concerned, I have asked you to tick on all these tools. So the license is valid for everybody for next 14 days, depending on your usage, your interest, how efficiently, how seriously you're using, we can think of extending your license. I promise it in front of organizing team. Next one, gene talks is done. Now gene AI. This is something you might not have seen anywhere. Access chemistry model. What it says, it can give you a route of synthesis. Suppose here I'm giving you job name. You'll have to give input. Job name. Say for example, I have to synthesize 1,5-diaryl pyrazole. So if you know some reference paper that this can be synthesized using substituted acetophenones, then hydrogen hydrate. I don't know reaction conditions and all. What product you want? You want substituted 1,5-diaryl pyrazoles. Am I clear? Can somebody respond?
Yes, sir, you are clear.
Okay, now we can give inputs that I don't want, many steps, synthetic steps. I want something which can be easily synthesized in 2 or 3 steps. Here I'll give you 3 steps. Complexity should be simple. Additional requirement, predict solvent, reaction condition, etc. And just like say generate synthetic scheme. It took few seconds. Still working on 20, 30, 40, 50 seconds, 60 seconds. It is searching all literature available on internet. The machine learning the model is trained using AI and machine learning tools. So it will fetch all the details in a simplified manner, available on open access or in the form of abstracts or in chemical abstracts, SciFinder, all the journals. In the meantime, I can show you some previously predicted reports. I just inform my see such kind of report it is generating. This was sent to our group. Job name was given. Substituted acetophenones and hydrazine added orthophenylene diamine. These were the reaction conditions I gave input. Created today, day, time, generated synthetic scheme. Okay. Here it is giving you details. Summary. Step 1 मध्ये काय होतं 4-methyl acetophenone hydrazone. Intermediate is so and so. Starting material is so and so. Step 2, you get a product summary. This can be generated, and then here it is giving you reaction followed by reaction condition. Reflux 80 to 100 degrees Celsius. Solvent is ethanol. Catalyst reagent, a few drops of glycyl acetic acid for 2 to 4 hours. Atmospheric pressure, normal reflux condition. Reaction mechanism is also predicted. Then this is second step. Substituted acetophenone hydrazone can be reacted to give the product. And these are the possible reaction mechanisms. So this is so good. If I say 1,5-diaryl pyrazole, ये हमने अभी predict किया था. Has given the results. Steps, reaction condition. Yield is also approximately 85%. This is a very good help to all the synthetic chemists, especially medicinal chemists, who are good at designing the molecule, predicting its ADME, toxicity profile, bio prediction, toxicity prediction, everything. But when it comes to synthesis, we face a lot of problems. If we get some inputs like this, we can definitely optimize the reaction and complete the synthetic process at the earliest possible without much more, headache, hesitations. So these are all advantages of GENI AI tool. Here you got one more tool, that is formulation. This is for pharmaceutics people. You can generate new title, and how it works, you have to enter formulation type, drug name, category, or AI generate multiple innovative research titles. आप टाइटल भी बना सकते हो, प्रोडक्ट बना सकते हो. You can predict its formulation also. Research paper, title, drug active ingredient, category, additional requirement. It will help you to prepare the title and predict its biological property. This is formulation AI. Again, for pharmaceutics people, you will have to register separately for this by adding your email address and password. Then it will take you to the formulation AI site, formai.ge-infos. What sort of formulations you can predict here? Nanoparticles, nanoemulsions, hydrogels, transdermal patches, and you have got excipient database also. Say, for example, we'll try to prepare some nanoparticles, start prediction. So these are the inputs you should be ready with: particle size, PDI, zeta potential, encapsulation efficiency, drug loading, percentage yield, and all. Am I clear?
Yes, sir.
Okay, thank you. So you have to simply select polymer. Say I'll select randomly. I'm not an expert of formulation development. I'll just giving you demonstration. You can try at your end, having background information. One category, one subcategory, then molecular weight. You can define the ratio 50-50, 65-35, 75-25. Polymer concentration you can randomly change. You can select any drug which comes under, I suppose, class 2 or class 4, BS class 2, BS class 3 solubility profile case of say. Suppose here we are getting taking doxycycline. Okay, we'll take curcumin. Its bioavailability is less. I said apply drug. Then आपने curcumin ले लिया. Synthesis method you can select stabilizer. Poloxamer, polyvinyl alcohol, Poloxamer 188, 407, twin 80, TPP, stabilizer concentration also you can change. Which method of synthesis you want to prefer? Nanopart precipitation, single emulsion, double emulsion, water oil, water system, or emulsion diffusion, ionic dilation, desolvation. Anything you can select, define temperature, stirring time, predict properties. Now it is predicted. These are
Sorry
all
to
possibilities. Yes, sir.
I have a question on synthesis approach, which you
I
just
think
have.
you are logged in with two systems. एक system को आप mute करो तो
Yeah,
noise कम हो जाएगा.
yeah.
Yeah, and can you be a bit louder, sir?
Sir, which algorithm work behind the retrosynthesis output in this web server? Which particular algorithm you are using?
Which algorithm we are using, right? So that
Yeah.
information we can get over here, sir. If you go to
Okay.
formulation AI, it
Okay.
gives us total information about kind of algorithm we are using. Advanced ML tools, okay? Comprehensive database, real-time prediction optimization engine.
Sir, is there any article recently published with this retro approach?
Yes, sir. I will provide few research papers published using these tools
Okay.
because
And
without that, it is very difficult to believe in or trust on
yeah,
any of
yeah.
the new system.
Sir, what could be the accuracy for this web server? Like, uh, this is just a kind of prediction with some algorithms. So, how much we can success with this scheme which is derived with this web server? Like,
You're talking about formulation development or you're talking in
yeah,
general, sir?
retrosynthesis, sir. Synthesis.
Retrosynthesis, sir, to be very honest, we have not yet tried, but I have just tried to predict the kind of compounds
Yeah.
which I have already, done synthesis of. So
Okay.
I can say around 90 plus percentage accuracy it is giving. Again, it depends. I've given a very general term, substituted pyrazole, right? Which type of substituent, depending on that, the reaction kinetics and regioselectivity and possibility of substitution at those particular reactions or sites that will vary. If you give specific input, probably it will give you a very clear idea because this is taking help of all name reactions reported we have learned in retrosynthesis concept.
Sir, is there any limitation for this web server that perform the synthesis? Is there any
Sorry,
limitation?
can
Like,
you be
sir,
louder, sir?
can we synthesize, can we develop a scheme for the synthesis of any peptide molecule using this web server?
Yeah, we can try it.
Okay.
Because it is taking help of all information available on the internet, and we are given the inputs of name reactions. To be very honest, this particular tool of retrosynthesis scheme I have tried today itself. So validation is in continuation. लेकिन try कर सकते हैं. If you face any problem, we can discuss in sufficient depth also, sir.
Okay, okay. Thank you so much, sir.
Okay. Yeah, as far as other tools are concerned, admit prediction, toxicity, docking, I have used those, for so many times. If you see, we have that database available with us also. Here I'm reaching to G-Inforce site. Okay. If you go to admin dashboard, I'll show you some predictions and how many people have used. All the details are documented over here. So tools usage statistics 674, ADME prediction, bio prediction 491, toxicity prediction 423, mole prediction so and so. So these are all analytics available on website. So people have preferred prediction of ADME and gene AI formulation. After that, toxicity, and then next one is this green wala bio prediction. Here we have all the documented details of which college, which students have used, usage count, percentage. I'll post the list of publications published on the WhatsApp group itself, sir. I think yesterday only I have sent some papers. Shall we go to next one? Gene mall is done.
Yes, sir.
Gene AI. Okay, Synthon doc. So this is something very interesting for pharmaceutics people. Synthon doc. Okay, here you can dock the co-crystals. Say, for example, you want to improve solubility of, I will say, ibuprofen. It is giving you ibuprofen, and you can select any of, say, amide or carboxylic acid or amino acid. I'll say alanine. Okay, so we have selected ibuprofen and alanine, and then just click on use this, use this. You can add name of the co-former, then say docking. So it will show what type of interactions are being, happening between a co-former or a solubility enhancer and your drug molecule. Whether it is, forming hydrogen bond interactions, hydrogen bond donor acceptor, electrostatic interactions, or pi-pi stacking interactions likewise. Here you will be able to visualize that these are the two molecules and they have such kind of interactions. It will give you idea about the bond distance also. Okay. You can select whether your drug molecule is to be presented in the form of the wire form, line form, or ball and stick, or its color should be based on element. It will give you idea about hydrogen bond, context, distances. If you want label, it will label whether the atom forming interaction is oxygen, nitrogen, or something else. Okay, it will show you surface that will give you idea about the possibility of solubility or solvent accessible surface area. And then you can export it in the form of result. I'll say download result. It will give you result in the form of PNG image. If you want it, to be incorporated in research paper. So this is really helping us, to interpret which co-former, which polymer would help in, assist in improving solubility of the less soluble compound. So this is Synthon Doc, a very good tool for pharmaceutical formulation scientists who can download the results. It will give you results in the form of zip file. You can unzip and explore all different interactions. Here it will give you result summary. Alanine, 1:1 ratio, energy is so and so, score is so and so. It is forming interaction with ibuprofen. Total co-formers are 1. If you are a formulation scientist, you will have a better idea about how many polymer molecule are to be selected and how what type of interactions you want, etc. etc. I hope this is clear. Now we'll go to last part, I suppose. You can select any molecule, any co-former, any ligand, and keep on trying. Only thing is, Synthon doc, you'll have to register separately because the script is different, the protocol is different, the learning, machine learning algorithms are different. So it is made available on separate panel. Now we'll go to next. For ADME we have seen, we just want gene doc suit. Synthon doc is done. Paraphrase humanizer, these are all, AI tools required for I'm not registered for this. You'll have to register, and then it gives you idea about AI similarity. You can sign up and start predicting whether your research manuscript is. I mean, you can check the plagiarism and all. Okay. So we are trying to give end-to-end solution, right, from prediction of active bioactivity of any molecule to its admit prediction, toxicity prediction, then its drug-like bioavailability, pharmacokinetic profile prediction, molecular docking studies, and whatever results you are getting, you have to write a manuscript. Even that manuscript is whether it is copy pasted, plagiarism, how much plagiarism is there, and how to remove that plagiarism. We have a humanizer part also. You have to simply copy paste the tool over here. Say, for example, I'll open any of the synopsis material. Okay, we'll take this part only. Just copy paste over here and then paraphrase it. You can first detect using AI detector, and then if at all similarity is found, detect AI. Nowadays, whatever research paper do we do communicate. Usually, it has to be screened using plagiarism checker softwares, and even AI reports are asked by some of the publishers. AI similarity report or It should not be beyond 10% at least. 10% margin is given just to just considering that some of the technical words like EGFR, name of the chemical, name of the method, these cannot be replaced. But anything which is copy pasted can be should be analyzed, and then you can take help of paraphraser and humanizer to reduce that similarity, to reduce the plagiarism. This is what this particular tool is all about. You can try it on your own, let it run. In the meantime, we'll start with Gene Doc. I hope every one of you have already registered this particular. So don't use this online available. This is older version. I have given instruction to everyone that you have to download the software, and then this is for those who could not download it. Then you have to click view my license for free trial. You have to keep ready this license key and activation code. Okay, this will be valid up to February 11, 2034. Others will get validity for 14 days. Fine. Then you can download the software by clicking onto this particular site. All right. Then you have to simply keep on clicking next, next, next. The software will be installed on your system, provided your antivirus software will allow it. Now I'll give demonstration about GeneDoc. So once the software is installed, you can click on the shortcut created on desktop. Is my screen visible? Hello? Hello, hello.
Yes, sir.
So, GeneDoc suite, this docking software will open like this. What you have to do is you have to load receptor either in the form of PDB or PDBQT file. Even if you write PDB code over here, it will fetch that from RCSB site. Then add ligands, define binding site, configure setting, run docking, analyze results. Everything can be completed within few minutes, less than 10 minutes. I'll give you demonstration. Load receptor. Okay, let us get started. I'll click on load receptor. I'll click on download from RCSB. I'm giving you 6LUD. This is triple mutant EGFR receptor. It has fetched the receptor. Total chains 1, 2, 3, 8, 8. साईट एवढं एवढं आतापर्यंत आरसीएसबी साईट आणि त्याच्यामध्ये सर्च करायला जेवढा वेळ गेला असता within fraction of seconds it has fetched it. Now we have to select chain A, or you can say select all, prepare protein. Now for preparation, these are the by default gas ginger Coleman polar only. You can select hydrogen only, polar, all hydrogens, or none. So I'll keep default. You can change the protonation pH the way you want. Remove water molecules, remove heteroatoms, merge non-polar hydrogens, remove non-standard residues and all. As default, like मैं अभी इसको protein preparation करो. Protein is prepared successfully. Just to 1 second. Now we have a protein. This is a protein. You can visualize. If you see, we don't have co-crystalline ligand over here because we have given a command remove heteroatoms. तो यहाँ से heteroatom containing ligand is removed. अभी आपको ligand docking करना है तो you have to simply add ligands. You can select the file already stored. I'll click on add ligand files. We have tried some ligands over here. OS1, OS2, and then osimertinib also. Let us take two ligands. Open, you can take multiple also. Just click on here, you will select, OS1 and OS2 ligands are there. 36, 39 bond size, status valid means structure is correct. Add hydrogen, we have already selected these generate 3D coordinators because if we input smiles, we'll have to generate 3D coordinate. Energy minimization, you can change force field, MMFF or UFF or chemical, whatever you want, you can keep the side. Check and fix valence errors. Prepare ligand. The ligands are prepared in fraction of seconds. Okay. Grid configuration you can define. I'll say based on co-crystalline ligand. Okay. If you want validate, it will take more time for validation. Validate means co-crystalline ligand will dock first and then again it will redock the same enzyme at the same site where co-crystalline ligand is docked. So अगर एक किक आपने click किया तो you can overlay co-crystalline ligand and newly docked co-crystalline ligand and check the changes in configuration. Right? This will take little more time. Do you want me to take this? अगर ले सकते हैं हम लोग.
Yes, sir, we can.
Right? We have taken validation. Now apply grid. Okay, these are all grid parameters. Scoring function is Vina. Exhaustiveness is 8. Modes 9. It means 9 times it will keep on repeating docking part. Energy range we want 3 kilocalorie. CPU and padding on all parameters. Apply grid. Grid applied successfully. If you see here, the grid is applied. You get 3-dimensional rectangular grid. Now you can run docking. %. First compound is getting docked. It has progressed to 10%. Here it will show you better base score. None of the program is so faster. We have seen we use Schrodinger's license copies since last 3 years. We have tried Autodoc Vina also. We have tried Molsoft and other tools also. Since this is AI enabled and it is already using Autodoc Vina as main algorithm, but all the process, you did not give any script, define anything while carrying out docking. Everything is AI enabled. And Rahul sir, you asked some questions regarding algorithms. I'll show you algorithms. It is already available under blog section of GE Inforce. We have Vina, Vinardo, and Autodoc. All three algorithms based on our choice. We can select any of the algorithm. So here we have results. Base score is minus 8.24 for first compound, minus 8.42 for second compound. If you see here, OS1, we have got 9 base poses. Which one is the best? More negative score, minus 8.241 is the base score. If you double click on this, I'll close this. You double click on this, it will open here. Okay. The molecule is docked. You can visualize it. You can go to interactions and check show interactions, show specific contacts. Which contacts you want? All contacts, hydrogen bonds, weak hydrogen bonds, hydrophobic, ionic salt bridge, pi stacking, cation pi, hydrogen bonds, metal coordination. The way you want, you can select. Accordingly, it will show the contacts. I'll say all contexts. So now it is giving all contexts along with amino acid, its residue, the table. Okay, next one is if you want to change, show specific, okay, interaction table, it will show you interaction table like this. Leucine A718 amino acid is forming hydrophobic interaction with the bond distance 3.56. Phenylalanine, usually we have to prepare this manually in any of the softwares. Here we are getting ready-made to, interaction table. The color code is indicated at the bottom of this. If you want to export this to CSV, here I'll show interactions of what? OS1 with 6 LUD. Okay. So this will be exported to Excel file. Yeah, export. We are not using this. Yeah, it has been, it is open available. OS1 6 LUD. So same table, distance, category, type of interaction, it has exported in Excel form. Okay. Then about analysis of all other, I'll close this. If you want to see receptor color, ligand color, you can say ligand color can be changed based on element, atom index, residue name. I'll say element because we want to map which atom is forming which type of interaction with receptor. Here you can go to view and then receptor color, chain, index, residue name. I'll write residue name. You can convert it into full screen. Anytime come out of it. Escape. Binding site. If you go to tools, here based on, sorry, here I'm getting into tools, then 2D interaction plot. It will give you 2D interaction plot. Yeah. Amino acid number, atom number, what type of interaction, and then everything is shown. You can export this in the form of PDF or export PNG. If I say export PNG ligand interactions OS1 with 6 LUD, we'll save it in the same folder. Rahul. Rahul should start. Right? You can click on any of the ligand and then check its interaction. Then zoom out, rotate this, reset its. That is all about 2D interactions. Show specific contacts, show ligand surface, interaction table, and then ligand You can click on labels. Receptor style also you can change. Cartoon, ribbon, ball and stick, licorice, line. Suppose I'm selecting line. Now it has simplified. Okay. View, receptor style. If I say backbone, it will be shown like this. If you go to view, go to ligand, ligand style, space field, like this it will. You can keep on exploring all these possible features. So it is none less than any of the paid software available in the market. Then view, receptor color, you can select based on chain, residue name, secondary structure, atom index, hydrophobicity, element. You can keep it uniformly blue just to avoid distraction. We are interested in the amino acids, its number, and then type of interaction or bonds. So you can change its view. You can keep it as aligned receptor, very plain, anything. So all these results are automatically stored in the folder, right? These are different poses, 9 poses for first compound, and then other compounds, 18, total 18 poses. You may get idea about RMSD also. And when you go to results, open result, phase 1, here result score you are getting, and these are all different complexes. Here in the form of this text also, mode 1, affinity score, that is docking score. This is RMSD. You'll find RMSD 0, 2.9, 5.6, 5.6, 4.8, 4.27. So everything which is required for proper interpretation of docking, you can make use of. So that is all about all different AI tools and their usage applications. I want every one of you to carry out at least one docking with the kind of target you have with you and ligand with you. I suppose we have some time. It is only 4:44. So we have got 45 minutes. You can try hands on and do ask me if you face any problem, any difficulty. In the meantime, I'll show you the algorithms to those who are interested. The scoring function we're using are these: Vina, Vinardo, AD4, Autodoc 4. The score, how the score is calculated, everything theory part is given here in the block section of geindoc.geinforce. I've clicked on block part. Right, these are different scoring functions, and this is the comparison about Vina, Vinardo, and AD4. Vina is faster. Vinardo is also faster. Accuracy is good in case of Vina. This is better for ranking. If you are generating new library of molecules and you just want to rank of these, say, 100 molecules, which are best 10 compounds. In that case, I would prefer you to use Vinardo for screening. Okay. Then we can go ahead. Remaining, the shortlisted 10 compounds can be docked using VINA tool, VINA algorithm. And then again for detailed energy, optimization, we can go for AD4. Then electrostatics, here we are taking in, VINA takes into consideration implicit, and here it is taking into consideration explicit electrostatics. Desolvation is both implicit here. Virtual screening, lead optimization, and energy analysis. For virtual screening, you can use VINA, and there are different scoring functions. And VINA scoring function uses combination of knowledge-based and empirical terms. Okay, विनाडो मध्ये कुठलं कुठलं वापरलं जातं, these are all details we have given. Which scoring function should you use? Performing initial virtual screening of large libraries, go for VINA. You want large results with good accuracy, go for VINA. Comparing results with published literature, go for VINA. Vinado is used for ranking for better correlation with experimental IC50 as to dissociation constant values. Synthesis based on predicted affinity. Likewise, these are the practical tips we are given, and these are the further suggested reading or literatures. Here we have beginner's guide, tutorials, case studies. What are different advantages and disadvantages? Updates and news are mentioned over here. Documents are also available. Release notes, features, workflow, file formats, video tutorials, FAQs, support, how it should be cited. I think sir has asked some references of already reported papers. I'll send across those papers to this particular group. These are all research papers I have already sent. I'll just pin the messages. You can see all above papers above this reference, sir. Any more doubt, queries? How many of you have tried? Oh, I'm sorry, there are 29 messages. I'll just check all the messages. Have I lost? No, the screen is visible.
Hello, sir.
Yes, sir.
Once you stop sharing, you will able to see the messages.
Yeah, I could come back to. I'll get back to these questions, sir. retrosynthesis algorithm. As I told you, uh, this was released very recently. Okay. Then what will be the accuracy for this scheme? Okay. About this retrosynthesis, all these questions will be addressed faster. I'll immediately post this. Next question is, which visualizer AI algorithm used in document server? This I have also answered, sir. Vina and Vinado. For
Yes, yes.
visualizer, we really need not have a very high graphics card and all. The simple laptop, Core 5, i5, i4, i5 would do.
Sir,
Protein preparation, ligand
uh,
preparation tool.
sorry to disturb you, sir.
Yes, sir.
I mean to say whether you are using JMOL or PyMol as a visualizer in your web server.
Yeah, I'll just inquire. What algorithm was given input at the back end, because that runs in the background. We are interested. I mean, I never went into details of all this because we are application scientists. Talking karo, result ले लो, paper publish karo. If I am I keep on digging out in details of algorithm, so probably कुछ application हो ही नहीं पाएगा. But still, I inquire and let you know, sir. That is, the developer would definitely be able to answer this. What difference between synthon doc and gene doc? Gene doc is for medicinal chemist point of view, right? For small molecule, receptor, proteins, targets, PDB files, and gene is a synthon doc. That is for two small molecules, maybe a drug molecule and a polymer, a drug molecule and coformer. Here, two dot mole molecules or two molecules in the form of dot mole format can interact with each other. Usually डॉकिंग के लिए हम लोग एक ligand molecule रहता है dot mol dot SDF and another is PDB. So here both the molecules are in the form of dot mol or dot mol 2. तो उसमें हम लोग interactions find out कर सकते हैं, especially for formulation scientists. So synthon doc is meant for formulation scientist and gene doc for medicinal chemist, pharmacologist, and other drug design team members. I hope, Ashwini madam, it is clear to you. Is it possible molecular dynamic study? Not so far. We are working on this, but right at this moment, it is not possible. Okay, is it considered water molecules during docking protocol? Rahul sir, this is a huge, uh, debatable question. We already have discussed this so many times on our CAT group with presence of Vijay Masan sir and so many people. Some people prefer docking using water molecules. Some do not prefer. Some believe that we are trying to simulate everything that happens in body. So body में receptor है तो वहाँ पे water molecules होने Hey, so why not take water molecules while carrying out docking? But some people do have, lot of objections or they have raised the challenges because of water molecules, proper intramolecular and intermolecular interactions हम लोग map नहीं कर पाते. There are, disturbances or unnecessary interactions intramolecular होते हैं. So it all depends on everyone's, applicability domain. But I never have checked वाटर मॉलिक्यूल लेने हैं या नहीं लेने हैं। अगर सॉफ्टवेयर डिजाइन किया है तो उनके पास बहुत सारा एल्गोरिदम, बहुत सारे कंप्यूटेशनल साइंटिस्ट रहते हैं। इट इज देयर लुकआउट टू गिव अस अ ऑप्शन। वाटर मॉलिक्यूल्स लेके करके देखते हैं क्या रिजल्ट आते हैं, विदाउट वाटर मॉलिक्यूल्स करके देखते हैं क्या रिजल्ट आते हैं, एंड देन गो इट। So it all again depends on what type of receptor we are using, what sort of interactions are taking place. Are these in situ water molecules present जो है, are these really hindering our results, creating negative impact on our results? Accordingly, we should take a call. That's what I honestly feel. So so इसमें बहुत सारा डिस्कशन हो सकता है। I am sure that you may not be really happy with this much of answer. Next one.
Yeah, sir. Actually, it varies from software to software. Basically, the Vina module do not consider the water molecule, whereas MOE consider the water molecule. Whereas in Schrodinger, we have to remove the water molecule within 3 angstrom of the selected cavity. Means it recommend not to put any water molecule in the grid box. And we have the docking without water molecule in Schrodinger also. So most of the software not recommend to use water molecule, and some softwares are there like MOE and some other like NRG suite. Okay, they recommend to add water molecules.
Yeah,
So
honestly
it is
speaking,
still
sir,
in people still are in dilemma whether to use or whether to skip the water molecule.
yeah, absolutely. I told you it's a big topic.
Yeah,
A lot
yes.
of debates have taken place. But let me tell you my experience. Published at least more than 10, 15, 20 papers wherein we have performed molecular docking studies. None of the reviewer has ever asked कि आपने water molecules लिए थे तो क्यों लिए थे, निकाल दिए तो क्यों निकाले. Never ever. I suppose my publications are published in all high impact factor journals. Definitely not without impact factor. 0.51, 1.52, 4, 5 इतने सारे हो गए हैं. But never ever the query was asked by any of the reviewer at initial stage, second, third, fourth stage कि water molecules आपने निकाले क्यों नहीं या फिर क्यों रखे हैं. I mean, nobody has asked us to justify. So I suppose कि ये शायद इतना important question नहीं होगा. As far as software companies are concerned, they are all selling thoughts to us. हम लोग जो available है, जो अपने best है, वो अगर लेके काम शुरू कर दिया है, and if this is accepted in scientifically renowned journals, reputed journals of high impact factor which are Scopus indexed, which are indexed in other well-known indexing agencies, Web of Science and all. So I never really gave it a serious thought, but definitely इस पे डिबेट हो सकता है. इतना सारे लिटरेचर अवेलेबल है, इतने सारे पेपर्स भी पीपल आर पब्लिश विद देयर ओन पर्सपेक्टिव्स. बट आई नेवर वरीड, आई नेवर टुक इट सीरियसली कि जो है अवेलेबल रिजल्ट जहां से अच्छे आते हैं वो हम इनकॉर्पोरेट करके पब्लिश कर दिए हैं. इवन वाइल गेटिंग द पेटेंट ग्रांटेड वहां पे भी किसी ने नहीं पूछा. तो शायद इसके लिए आई हैव नेवर given it a serious thought. Of course, the question is important because some software are saying कि ये करो, दूसरा बोल रहे ये करो. So to be honest, I never emphasized on this particular topic.
Yeah, thank you so much, sir, for your detailed analysis and your detailed information on every aspect of the tools which is available in G-Infos. Thank you so much, sir.
Yes, sir. There are a few questions. Is it possible to get zero RMSD in docking results? I have never witnessed zero, but yes, we can reduce RMSD to 0.5, 0.6, 0.7. यहाँ तक तो अभी तक आए हैं. अभी तक we have never witnessed zero RMSD. Probably we'll have to check it. What is the difference between? Yeah, that is addressed. Okay, any more questions? I think Rahul sir, Dr. Rahul sir has maximum number of queries that he has helped every audience to get answers to all these queries. और किसी को कुछ doubt रहेगा तो we can address, otherwise all yours sir.
Thank you, sir, for delivering such an informative and engaging session. Your expertise and insights have gratefully enriched our understanding, and we are extremely grateful for your time and contribution. If anyone have question, they can unmute.
Okay, I would request organizers to kindly share the feedback submitted by audience, and especially the kind of questions we are going to ask them in the form of assignment. So
Yes,
based
sir.
on that, we'll definitely come to know how far we are successful in reaching to audience at large,
Yes, sir.
and
We
what are
are
the
providing.
points wherein we need improvement. All the suggestions, critical comments are most welcome, because I believe in continuous learning. I never said ki I know this and I'm complete, and that nobody should have that approach. Thank you. Thank you very much.
Before moving ahead, let me summarize you this session. This session on AI-based ADME in drug discovery highlighted the transformative role of artificial intelligence in pharmaceutical sciences. The session explained how AI-driven ADME prediction enhanced the efficiency of drug discovery by enabling the faster screening, accurate predictivity modeling, and data-driven decision making. It emphasized that artificial intelligence significantly reduced development time, cost, and failure rates by identifying the potential issue early in the drug development process. Overall, the session provided valuable insights into how artificial intelligence is revolutionizing early stage drug discovery and improving the success of novel therapeutic candidates. Now, we come to the end of the session, and before formally conclude the session, I request all the participants to switch the camera on for screenshot. Thank you to all respected dignitaries, esteemed guests, faculty members, and our dear participants. On behalf of the organizing committee, I extend my sincere and heartfelt gratitude to our esteemed speaker, Dr. Shashikant Bhandari sir, for delivering such an enlightening, insightful, and informative session. I would also like to express my deep sense of gratitude to our honorable management for their constant encouragement, guidance, and unwavering support, which made the successful organization of this program possible. My sincere thanks to our respected principal sir for their visionary leadership, continuous motivation, and invaluable support throughout the planning and execution of this event. I extend my heartfelt appreciation to all the faculty members, organizing committee members, and coordinators for their dedicated efforts, planning, and teamwork, which ensured the smooth conduct of this program. Special thanks to the technical and support staff for their prompt assistance and efficient handling of the technical arrangement, enabling the seamless online delivery of the session. Finally, I would like to thank all the participants for their enthusiastic involvement, active interaction, valuable presence, which truly added value and success to today's program. Finally, I thank all the participants for their active participations and cooperation. And I request all them to attempt today's assignment and provide your valuable feedback. Before Concluding, let me tell you about the pre-cap for tomorrow's session. Tomorrow, we will begin with our first session on e-content and artificial intelligence for quality teaching by Dr. Prashant Argade sir, where participants will gain insights into developing effective digital content and the role of artificial intelligence in enhancing the teaching quality, student engagement, and learning outcomes. This will be followed by the second session on artificial intelligence in drug discovery by Dr. Somdatta Chaudhary sir, which will focus on the application of artificial intelligence in modern drug design, molecular screening, and acceleration the drug development process. With this, we come to the end of today's session of the online faculty development program. We sincerely thankful all the participants and speakers for the valuable time and active involvement. We look forward to your continued participation in the upcoming session. Thank you.
Can I take a leave? Dr. Rahul sir, madam?
Yeah, sure, sir.
Okay, thank you. Good day.
Thank you, sir.
Okay, thank you very much.
Hello.
Yeah, just a moment. I'm
Hello.
copying the
Sir,
query.
Sir, thank you, sir. From the organizing team, I want to thank you once again. Actually, you were very enthusiastic, like sharing all the app which you have developed and logging in all the participants. Like you were active for long hours on the WhatsApp in morning also we saw it. So it was
Yeah, right from
quite
yesterday.
appreciating, and we are really thankful to you, sir. So I extend my warm thanks from all the organizing team and both the institute for your active insights which you have shared with us. Thank you so much. Thank you so much, sir.
Most welcome.
Yes, sir.
Okay.